The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a distant location. The encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then, in case of translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled by either permitting or blocking access to the site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then, during elongation, the RNA polymerase slides along the template DNA strand. As the complementary base pair up, the RNA polymerase links nucleotide to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator region of the gene, the messenger RNA transcript is complete and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein and non-coding sections called as introns. In order for the messenger RNA to be used in translation, the non-coding introns needed to be removed and modifications such as 5' cap and a 3 poly A tail are added. This process is called intron splicing and it is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nucleopore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translate into a protein? The nitrogenous bases are grouped into 3-letter codes called as the codons. The genetic codes includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, one that codes for start codon and 3 that codes for stop codon. Translation begins with the messenger RNA strand binds to the small ribosomal subunit upstream to the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule bind to the start codon, the larger ribosomal subunits binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are three distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the mRNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site and a peptide bond forms between its amino acid and the one attaches to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now charged transfer RNA molecule exits from the E site and the A site is opened up to accept the next transfer RNA molecule. Elongation will continue until a stop codon reaches. A release factor binds to the A site at a stop codon and the polypeptide is released from the transfer RNA to reach the B site. The entire complex dissociates and can resemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. The modifications takes place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestine, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, and secreted during a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. The proteins are needed for most physiological functions of the body to occur properly, such as breaking down of food particles in digestion, and the process of transcription and translation makes the production of proteins possible.